Hi everyone, my name is Kip Ludwig. I am the co-director of the Wisconsin Institute for Translational Neural Engineering, and I'm happy to be here today to talk to you about a minimally invasive neuromodulation through an in-body curing injectable electrode, or something that we like to call the Injectrode. Here are my disclosures. I do a lot of preclinical work to help companies both doing non-invasive and invasive stimulation. Perhaps the most germane to this conversation, though, is I am a co-founder of Neuronoff. That is an entity that was created specifically to attempt to commercialize the Injectrode concept. So there's a wide variety of electrode concepts for neural stimulation. They range from being very non-invasive to invasive implants where cuffs are placed around the electrode, and in some cases, any variant in between. In some cases, you could have a percutaneous connector going to an outside power source that's then stimulating the deep nerve. You can also have a chip that is wirelessly powered and wirelessly programmable that's placed surgically upon the nerve as well. And so there's really good uses for non-invasive and for invasive techniques. Non-invasive techniques to stimulate the nerves can have some fundamental limits associated with them. You can defibrillate the heart from the surface of the chest, but it's really hard to do that without stimulating every nerve, muscle, and gland in between. So this is a great paper by Seth Hayes where he was doing vagus nerve stimulation, which is something that I'm very interested in. Vagus nerve stimulation can change breathing rates and it can change heart rates. And what he showed in the rat model is that when stimulating invasively with a cuff wrapped around the vagus to activate what's called the Herring-Brewer reflex, which actually changes breathing rate, he only needed 0.4 milliamps. But in a rodent where the surface of the skin is 2 millimeters away, he needed 34 milliamps to actually non-invasively get a breathing change in the vagus. And it was impossible to do that without activating the neck muscles. In fact, the amplitude at which the neck muscles activated was roughly 1/15 the level needed to get some sort of breathing rate change. Now, I typically work in large animal models, and so I was interested to reverse this. And in this particular case, we stimulated the vagus invasively using the waveform that's typically done with a non-invasive stimulator. The gamma core system actually doesn't stimulate at a constant frequency. It bursts 5 kilohertz at a given frequency with the intent of effectively lowering the skin impedance. And so this is actually the heart rate drop that we got stimulating the vagus when stimulating with the normal Livanova cuff with a biphasic pulse versus the gamma core waveform. And you'll see that we're getting about a 20% reduction in heart rate when we are directly stimulating the vagus by placing electrodes on the vagus surgically, but with that waveform. However, if we started transposing just a little bit of tissue, so in a pig, the surface of the skin is much further from the vagus, so we tried to do the reverse. Once we got a good heart rate with a surgical intervention, we started just putting a millimeter or two of fat in between the electrode. And what you'll see is just putting a millimeter or two of fat, we no longer have a 20% heart rate drop, we have maybe a bare 1% heart rate, and we've also gone from 3 volts peak to peak to 4 volts peak to peak. Now as we crank it up, this is what that drop was when we didn't have any fat transposed on the far left side. In the middle was the drop we had when we just put 2 millimeters in and cranked it up a little. If we start cranking it up a lot, we get still only about a 5% heart rate drop. Now keep in mind this is only putting 2 millimeters of fat in between the electrode and the vagus. The actual situation in humans is 2 to 4 centimeters. Moreover, a bipolar electrode, the fall off of an electric field is 1 over r squared. This is an exponential function, so you would actually need several orders of magnitude more than this to stimulate the deep vagus from the surface. And the problem with that is you have superficial nerves like the great auricular branch and the transverse cervical nerve that are right above the muscle that you have to stimulate through to get to the vagus. So you wind up getting intolerable side effects before you get effects. So implantable devices have their own unique brand of problems or things that have to be overcome. They can be very useful in some cases. However, designing these is very hard. This is data from the follow-pace study as well as vagus nerve, spinal cord, and deep brain studies. And what you can see is that there's actually a high degree of complication with these implantable devices. They're complex. They have a battery and computer that's implanted in the chest. They're typically routed to where they're going to be stimulating. And they wind up with lead failures of all different sorts. They also wind up with supply chain failures. Roughly for the pacemaker, which has 900,000 implants a year worldwide, you wind up with a 12.4% complication rate within the first 2 months. You wind up with another 9.2% complication rate within the first year. Similarly, the more complex a device comes, the more you're going to wind up with problems in your supply chain. The vagus nerve stimulator has been around since 1997 and is actually, in just the last several years, the Sentiva system is on its 5th recall. And this is because if there's just a small problem in supply chains, it can actually create a big problem. And if you have dozens of suppliers, there's more likely to be one of these problems. So me coming from industry originally, realistically, I understood people who took great ideas from academia and tried to apply them to the neuromodulation industry. And the truth of the matter, it doesn't work really well. In industry, the one immutable fact is that you don't set your price. Insurance payers do. So you actually have to work back from the price you can get for a device for a specific therapy and its modality and figure out what you can manufacture with the supply chain to sustain the market. So I've been interested in flipping this since since I was in industry and thought about, do we really need a $40,000 pacemaker-esque system to do a lot of the things we want to do with neuromodulation? So my postdoc fellow James Trevathan, and in collaboration with Andrew Shostall from Case Western and Manfred Frank from Neuronoff, came up with the idea that maybe we can come up with something that's an alternative between invasive stimulation and non-invasive stimulation. And this is what we call the Injectrode. This is actually, to some extent, based off of FDA-approved surgical glues, which can be injected as a liquid and then cure within the body. And in this particular case, it's doped with metal particles to make it conductive. So a lot of the data I'm going to be showing you, because I come from industry and had a background in developing tests for invasive electrode implants, this is a paper you should be aware of where I tried to go over all the testing that we did for the CVRX electrode that stimulates the baroreceptors to submit for FDA IDE approval. And now this actual device, as of the summer, was PMA approved as well. So the Injectrode, because it's liquid in a syringe or liquid E, and then cures and becomes stretchable to some extent once it cures, you can actually inject it, say, in the carotid sheath to get the vagus, or on a complex structure like a plexus or foramen. It actually maintains its impedance when you stretch it as well. It has a Young's modulus of about 70 kilopascals and compared to regular electrodes, which are about 50 gigapascals. And so it can take a lot of stretching with respect to relative movement, especially as you get to large animal models. So in our original paper that we published in 2019, we had done a quick proof of concept with a silver-based particle strategy, but we've now moved on in recent days to a gold wire strategy. And what we found is, because the matrix for the Injectrode, this essentially polymer, is fluid permeable, the particles, the metal particles of Injectrode, fluid can get in and access all of that surface area on the interior, as opposed to just acting on a surface of a regular, say, solid wire. And so what we're seeing is, with a gold Injectrode, we're actually getting much more area under the curve compared to regular gold, which is orange. This is cyclic photometry for those of you who are familiar with. Platinum and your hydrogen peaks are in blue. And we get essentially a much expanded area for the gold Injectrode. Similarly, if you're going to look at the electrochemical impedance spectroscopy, the gold Injectrode compared to a Livanova clinical electrode that's platinum radium with the same geometric dimensions, you wind up with a couple of magnitudes order lower impedance. So this is promising, even though we have this particle-based strategy that to some extent may work through what's called percolation theory. So we've shown in our previous paper that when we put this on a nerve, either the brachial plexus, or in this particular case, we're directly comparing it to vagus nerve stimulation with the clinical electrode, we see that we can get the same heart rate drops at the same voltage. Because the sizes are different, the current can be a little bit different, and that's because the activation function is actually more a function of charge density than from current applied. So we've tried to extend this from directly stimulating these nerves with a wired connection to something we call the injectrode collector concept. Now in this concept, what we do is we inject the electrode with a syringe around the nerve, we extrude it back to the just under the skin, and we inject a bit more of it to make a collector. And instead of having a percutaneous connection that spans the skin, we actually take a TENS unit from Walmart and we capacitively stimulate it. So there's nothing spanning the skin here. But more importantly, the pulse generator, being from Walmart, is 50 bucks. So this entire system is actually quite inexpensive. There's only one part put in the body, which is the injected material that's cured. So it has many fewer failure modes. And so we've actually shown in a pig model that when we use this injectrode with the collector system, in this particular case, the collector is not in the neck, it's actually in the chest. The TENS unit is over the chest, and then it's been routed to the deep nerve. You can see that we can repeatedly get these red periods are periods of stimulation of the vagus. You can get nice heart rate drops. And then when we move the collector from the chest and we stimulate with the TENS unit, we get nothing. We've shown this in several animals at this point. So this is really a unique strategy to potentially get from this complex $40,000 implantable pulse generator and all of its parts and all of its complex supply chain and all of its failure points to really get to a one material system, and then an implantable pulse generator that's not implantable, that non-invasively stimulates. We're essentially creating a neural bypass from the skin to the deep nerve without stimulating the superficial nerve. And so this is modeling work done by Scott Lemke and Bobby Graham at the University of Michigan. And we've started to actually incorporate, as part of our Injectrode collector refinements, this collector that's superficial and models of the superficial nerves that would create your side effect. That's the big problem limiting the electrocore system, for instance, for non-invasive stimulation without a collector. They actually wind up getting superficial neck muscle activation to the point where they get lip curl, and then they can't turn the current up anymore, which pretty much prevents you from getting current to the deep vagus. We have shown that by using this Injectrode collector system, it's possible to get 2 orders of magnitude more current to the vagus without stimulating the superficial nerves. And now we're starting to use this modeling framework to optimize this collector system for robustness. Realistically, if you're going to have this applied by a patient in the home by themselves, they're going to have to be able to stimulate these collectors, and it's going to have to be relatively robust to mistakes. So we're really excited about the possibility of kind of changing this trade-off for a minimally invasive procedure, and doing so inexpensively and in a very simplified way so that there'll be many fewer failure points compared to a regular implant. So the obvious question is the gold injectrode biocompatible? We've done long-term rodent studies where we injected subcutaneously and then looked at histo and or gross morphology of the injectrode. At the top, you'll see the injectrode with a version of the matrix that actually reabsorbs, and you'll see revascularization actually through the gold particles. In the lower left and the lower right, as you zoom in, you can actually see there's very little scarring associated with the injectrode. So for the electrode wonks, I want to talk a little bit about voltage transients. For those of you who do voltage transients to determine the charge injection limits for biphasic pulses, you're going to see for platinum iridium electrodes, across the history of platinum iridium electrodes, the charge injection limit is actually widely varying. And we think we've run into an issue. The injector actually looks very good when we calculate the charge injection limit compared to platinum iridium, but we've also noticed a problem with maybe how historically voltage transients were taken. And so historically, voltage transients were taken with oscilloscopes doing 1 meg or 10 meg of input impedance. And the problem being, especially as you get to smaller electrodes, you wind up having appreciable shunt loss between the working electrode and the reference electrode. So this on the right is actually the DC offsets measured with a platinum iridium electrode when you have 1 meg versus 10 meg versus 10 gigaohm of input impedance. And what's happening, because current is shorting from your working electrode to your reference electrode, you're actually minimizing the anodic offset that you would get. And oddly, the anodic offset that you get helps you go to greater currents before you get to the cathodic limit and causing hydrogen evolution and or hydrolysis of water. So this is what it looks like when you're doing voltage transients and you're trying to get the electrode polarization. So before was kind of a zoom in of the offset, but this also shows you that your electrode polarization that you would get calculating it via standard methods actually changes a fair bit whether you have 10 gig, 1 meg, or 10 meg. The other interesting thing about having that current pathway from the working electrode to the reference electrode is you also change oxide formations on the surface of the electrode as well. So this is just something for us as a field to be aware of and might be an interesting panel question discussion. I'd like to thank my collaborators. I'd also specifically like to thank the NIH U18 program and the DARPA TNT program, of which the Injectrode was developed under. So thank you very much. I look forward to answering questions in the panel session, and you can also follow me on LinkedIn or Twitter.